In order to grow and to heal, our bodies need the ability to produce additional cells. Every cell has a cell cycle. Within that cell cycle is mitosis and meiosis, which is carried out by reproductive cells. So the cell cycle for our cells is on average 24 hours long. You will see different numbers in different places for different types of cells, and that's very common, not unusual at all. I like to grab that number 24 hours because it being the 24 hours of the day is something that we can relate to and remember somewhat easily. Every cell goes through a cell cycle. This is illustrating an entire cell cycle. This is from the time a cell is produced until it is going to divide again. The majority of that time is spent in interphase. Oftentimes there is confusing information released that interphase is part of mitosis. Technically, it is not. Mitosis, on the other hand, is the actual visible nuclear division events. In interphase, we have three separate phases where we have growth and synthesis going on. The first of the growth phases, the G1 phase, is carrying out the normal cell function. Cell is getting larger, organelles are duplicating, protein synthesis is occurring, whatever it is that that cell is supposed to be carrying out. Then we have an S phase here. During S phase, DNA is replicated. At the same time, we have to have histones for the DNA molecules to wrap around, so the histones will be synthesized at that time too. Then in interphase, we move into the G2 phase. During this phase, we see centriole replication and the final synthesis of proteins in preparation for our cell to divide. Looking very closely at mitosis, this may take as little as 15 minutes out of the cell cycle. In mitosis, we have four phases. The first of these phases is prophase. This is going to look really odd because I'm writing red on red. I did not think to change my pen color. The second phase is metaphase. The third one is anaphase. And the fourth is telophase. Another event that I want to identify at this time is cytokinesis. We'll talk about this a little bit more during anaphase, but cytokinesis is where the cell contents are actually separated. Focusing on prophase first. During this time period, we have a lot of things that are going to happen. One, the nuclear envelope is going to disintegrate. Apparently I can't spell. Okay? Secondly, the nucleolus is going to disappear. Third, the centrioles begin migrating to opposite sides of the cells. Pen was not cooperating. While the centrioles are migrating, there are contractile microfilaments forming between them that are called spindle fibers. The major event of this phase is that chromatin is going to coil up and become chromosomes. Chromosomes is a very familiar form of DNA for all of us. Unfortunately, it is not made known to us that chromosomes are not always in that form. During interphase, chromosomes are needed for, the DNA is needed for protein synthesis and so on, and we're not going to be able to access those genes if they're all tightly coiled together. Metaphase, in contrast, is a very simple phase. It has but one event, and that being that the chromosomes are going to line up at what we call the equator. Very simple. That's the extent of what's going to happen there. Anaphase also is quite simple in comparison to prophase. There is but two things going on. One of these being that the chromosomes are going to separate and begin migrating to opposite sides of the cell. The other thing that is going to happen here is that cytokinesis is going to begin. This is the process of dividing the rest of the cellular contents into two separate cells. This particular cell that is shown here is not constricting yet, but as these chromosomes move further apart, the cell membrane is going to begin to constrict so that it can eventually come together and form two cells. Our final phase is telophase. Telophase, again, is very busy. We are simply going to reverse the events that we saw in prophase. Chromosome is going to become chromatin. It's going to uncoil. Nuclear membrane is going to reform. Nucleolus reappears. And lastly, cytokinesis completes. You see that there are still a few fibers going across between the two cells here, but the cell membrane between them is nearly complete. There is an animation posted in Webliography called A&P Flicks. Please take time to see that if you would like to see mitosis in action. Let's compare mitosis with meiosis. The whole idea of mitosis is to produce body cells that are genetically identical to one another. The role of meiosis, on the other hand, is to produce genetically variable cells for reproduction. The second, or maybe I should say secondary goal of meiosis is to produce cells that have half the number of chromosomes that a body cell would have. During fertilization, the half from the male is going to bond with the half from the female, and we would not have a viable cell if we tried doubling that. What happens with the meiosis is that initially all of the chromosomes are doubled like this. Then they go through meiosis 1, which produces two cells with half of the number of chromosomes. Then these cells are going to go through another cell division event, meiosis 2. There will be no DNA duplication between, and we will end up with then 4 cells that have half of a normal number of chromosomes. During the process, there is some recombining of the DNA so that a chromosome or a chromosome that is in one cell might not be exactly the same as what ends up in another cell.